have to check in for a flight in 27 minutes. So my alarm will go off and I'll stop for a second. Very uncomfortable Allegiant flight. Let's see. So we're recording now, and I will get started. The last 2 days, I think maybe I did it twice, yesterday or Monday. I posted a couple of additional recordings because I had to leave rather suddenly. Just some stuff came up. I was here, and then I had to leave. So if anybody showed up after I had left, I apologize. Came on relatively suddenly. But nevertheless, I had covered, this is the pertinent to the sheep dissection, which we're doing today. This will probably take about an hour, maybe a half an hour or 45 minutes, and we'll do the dissection and we'll be done. For you folks here and for you not here, we're going to do the exam next week online. Everybody, I'm going to send it to you on or about the time we start. Normally about this, roughly this 12:30 nugget, and give you a couple of hours to complete it. There's going to be a written section with, multiple choice with about 25 questions, and then there's going to be, I'll make sure I have a study guide out just in case I don't. And then there'll be about 15 structures to label. So I'll send those, or 15 slides with areas to label on them. You can just mark those however you wish on it, or create a list with whatever your answers are to the questions, and or identifying the structures, and we'll go with that. Doesn't matter, very, very flexible. I just want to get everything in an adequate time for me to post the grades, which are probably going to be due toward the end of next week or early the week thereafter, because we do start the next 8-week session. So that's everything so far with that. I'll send a more detailed thing via email in case someone doesn't get around to reading this at the honor before that happens. So there's only a couple of really the only structures in here or aspects of the brain are the limbic and the reticular formation, and then at the very end of this we look at the brain coverage, which are really important, and that's the meninges. So the limbic system is very interesting. It's sort of, imagine deep in the center of your brain, surrounding the big cerebral lobes and the diencephalon. The largest structures is this series of combinations of gray and white matter called the limbic system, and it's all connected by something called fornix. And really, this is the big deal when it comes to our emotional aspects. It's how we recognize anger or fear or assessing danger, those types of things that are really there. This is what it looks like graphically. Probably one of the slides you're going to see on the final, without obviously the labels on it. Interestingly, sense of smell is a big player in that, and that's part of the olfactory, so your sense of smell goes into it. A lot of your memories are triggered by emotions or implanted by emotions. When you're upset about something, you're happy about something, you remember that stuff. And that has to do with something that's here on the inside or internal portion of the temporal lobe called the hippocampus. And then as you work your way up, in that same area with another piece of gray matter is called the amygdaloid body, and that's really the anger or rage center. When they do studies on people to look at those with anger management, a lot of times we'll see either irregularities or an excessive amount of stimulation of those areas. And we all know people who just very, very, really have anger management issues, no other way to say it. So this is part of it. Whether or not this plays a role, it may play a role in psychosomatic illnesses. A lot of times that affects people emotionally. They make themselves sick. I mean, most of us Some folks have a tendency to internalize that stuff, and it bothers them. I'm one of those, interestingly enough. So I just thought I'd throw that out there. The reticular formation is all about cortical arousal. It's what keeps you awake. So you have aspects in sight and in smell and in motion and in pain. Think of all the things that allow you, that effectively, they call it the reticular activating system. It sends impulses up to your brain. And, for the most part, let's say you're sleeping, you're taking a nap and the TV's on, or you're listening to some music at the same time, and you sort of filter it out. Your brain has that capability. But if someone slams a door, somebody turns on a light, somebody shakes you, or my dog jumps on me, which is a large dog, those are the kinds of things that tend to wake you up. And so you can see, you can have stimulus from touch or pain or temperature. You can have it from visual, auditory impulses, and they'll send signals up to the larger part of the brain for you to remember this. And really, the rest of this in here is not terribly, is much more physiologically based. That's why I'm going through it. I do this in, just a quick note about this. We do this a little bit in our advanced anatomy labs. Is I have a device that we can read brain waves. And for a very long time, and to some extent legally still is, the way we define someone as being dead or brain dead. You trace their electroencephalogram, which is a non-invasive technique. You just put sensors and they pick up electrical activity. It's a very readable pattern. And we can do that. It isn't, the device we have isn't anything like this or as sophisticated, but it can pick up impulses. And it's interesting, there's different ones when you sleep, different ones when you're awake. It's one of the ways we used to diagnose seizure disorders, which some people will call epilepsy, things like that, and different kinds of waves. And back in the old days, before we had advanced technology, when you had sleep disorders, a neurologist would read through these like a book, be able to isolate, look for tumors, look for sleep irregularities, all sorts of. I used to watch a neurologist do that when I was a student. Well, I was still a student. I didn't even have my doctorate. So that was the better part of 50 years ago. So the technology has certainly changed. And this is how we characterize different kinds of seizures. I do this in lecture. And all the different types of sleep, and I don't get into it that much, not an area of my expertise. And which brings us to the anatomical, the meninges or meninges, however you want to pronounce it. That's the plural, multiple layers. Menix is technically the singular. And it does just what it says here. It protects blood vessels, protects circulatory supply, has a specific fluid, cerebrospinal fluid, and to some extent separates area within the skull. The three layers you will need to know, because you'll see this diagram on the final. The dura, which means hard, matter means material, arachnoid because it looks like a spider web, and pia, which means soft or delicate. So this is the illustration that you will see. And effectively, and we're going to look at the one in the spinal cord. It's a little bit different. Okay? Here in the brain, so underneath the skull and the bones of the skull, you have a layer of dura, one against the bone and one against the brain itself. So periosteal against the bone and meningeal, which is really the part that covers the brain. They have little gaps. These are called sinuses. They're filled with blood and the recycled cerebrospinal fluid that comes up by way of these, which are called arachnoid villi or granulations. They're like little siphons to siphon off extra fluid. We make about a pint of fluid a day, we siphon it off every day. The fluid is stored here in what's called the subarachnoid space. So you have the dura, then you have the arachnoid, a much thinner layer, but with a large space, and there are those little strands that look like a spider web, and then the pia, which is directly adherent to the brain. You will see the dura, possibly, or aspects of it on this dissection. You will the pia is on the brain. It's just so delicate and part of it. If you try to lift it up, little bits of the brain tissue will come off. And then underneath is gray matter and then white matter. So they're natural dividers. I have to look at my specimens that we have here and see if any of them have the dura still on, so we can use that. And I mean, if there's only 3 of us, we can just do this as a demo, so you can see it. I can just handle it and show it to you. And so the dura, by its name, you and I, with a piece of Duramotor, could play tug of war. It's just unbelievable. Years ago, and I was telling my colleagues about that this morning, I was teaching a graduate anatomy lab over at Chatham University here in town, Shadyside, and we had a human brain and human dura, and you can't imagine that you can't how strong it was. So you have the 2 layers, and these little splits or gaps called sinuses. Dividers, which collectively are called forks, which you may or may not see, where they make a tent over the cerebellum. And this is part of the blood coming back from the brain, filters in these what are called sinuses, and eventually they sort of unite around here and help to form what are called the jugular veins. Carotid arteries bring circulation to the brain, jugular veins return it back to the heart. Kind of what it looks like. The arachnoid is delicate, the space is where the fluid is. Same illustration, the P is clingy onto it. And when you hear about meningitis, it means it's inflamed. It is inflamed virally and potentially bacterially. The bacterial one is a major, immediate life-threatening situation. The viral one is something that anybody can get it. When I tell you that, during your lifetime, inevitably you know somebody will have it, because it's much more treatable. My daughter, I'm going to tell you, maybe less than 15 years ago, had it because of it's like it was a herpetic, meaning a herpes infection. I saw a cold sore, rather than coming out on her lip or inside of her cheek, ended up creating meningitis. We knew it because we responded to the medicine, the antiviral medicine that was specifically for that. And the neurologist confirmed that she was a mess. I mean, she was discharged from the hospital, had to be readmitted. She was so bad. Thankfully, no long-term problems. The fluid is very much of a cushion because that brain is so heavy, it would literally plunge down into the foramen magnum in the skull. And so it's really, and again, it's sort of mostly plasma with some chemical alterations made in these choroid plexes. This is the circulatory pattern. So it exits in these places to surround it, can exit all the way to the bottom of the spinal cord. We'll get to that in a second. And that's about, and really, what's unique about here is called the blood-brain barrier. The capillaries do not have gaps that allow fluids like they do everywhere else in your body. These gaps are really not present. There's a lot of tight junctions and layers of the astrocyte connective tissues around it. Okay. Now, one more little presentation to go on this. I'll probably have to interrupt this ever so briefly to show you the brain dissection, which I will do. Okay, so this is the other part, and the last presentations I've either sent to you or I've posted have been mostly on, over here, I've been on the brain. This is the rest of the central nervous system. The spinal cord is considerably smaller but much longer. It's fairly narrow, has a relatively small diameter, wider at the top, shorter at the bottom. And interestingly, it don't go all the way down. So, because our body our bones grow faster than the elongation of our central nervous system. Effectively, what happens, the spinal cord starts at the base of the skull, at the foramen magnum, and ends between the first and second lumbar vertebrae. That leaves a pretty good piece of space, maybe a couple of inches, where there's really no cord. There are nerves. And that's very important anatomically because what it permits is a physician or an anesthesiologist to do a spinal tap, to be able to pull out cerebrospinal fluid, which is a really critical test, specifically and perhaps most commonly to just diagnose, is the diagnostic test for bacterial meningitis. And so this is a really critical area. Here's what the cord looks like. It's thicker at the top than down here toward the bottom. It ends about here. It's thicker. It has what are called enlargements at the cervical area because all the in and outs to the upper extremities are there, and in the lumbar area, because all the in and outs to the lower extremity are there. And it has spinal nerves coming off all of these. So we do the nerves separately. That's an AP2 thing. We start with really the information about, that's part of the peripheral nervous system. So we're just going to look at the cord. So the dura, it doesn't have gaps. It's got one layer. It doesn't have that layer, one against the cord, one against the bone. What surrounds it is one layer surrounded by fat. And that's an area where medication can be introduced for, let's say, a woman who's delivering a baby. A lot of times they'll give them a spinal type of anesthetic, but it's not the same thing as having a spinal anesthesia. Okay? They have, what you're doing is called an epidural, which means it's outside of the dura into that fatty layer, and it acts more to relieve pain than to create anesthesia, which is the absence of pain. And that's because there is an epidural space, which is much more easy to access from the back than in the front. Again, the fluid's in that subarachnoid space. And as you can see, this is the area between roughly L2 and L5, L4, L5 area where you can do this. Here you can see a demonstration. Someone who's very highly trained, typically an anesthesiologist, will insert that needle into that space, remove the catheter, and it allows fluid to flow out, and you collect 3 successive tubes to analyze. I have never done it. I've seen them, but I've never done it. Okay? Anatomically, the cord ends in a conical structure called the conus medullaris, which you should know. It has a thin filament called the filum terminale, or terminal filament. That's me reminding me to check in. And has little ligaments to kind of secure it called denticulate. They kind of look like little teeth. And other than that, you have spinal nerves coming out. And all the spinal nerves at the bottom, since they kind of look when they were teased out, and I could show you that if we had a human spinal cord, which I did years ago, the nerves look like a horse's tail, so they called it a horse's tail, cauda equina. And so you can't really, so here's what it looks like. Here you can see these little bits of nerve coming out, the thickness of the cord. As you work your way down, you can see it a little bit more how the nerves come off and go through the spaces. This is the bottom here. It comes to that sort of a tip, the conus medullaris. Here's the filum terminale. And on either side here are these nerves, the long nerves that are going down toward, your feet and your legs, that are called the cauda equina, or horse's tail. And that's really, the rest of the anatomy is fairly straightforward, and I'm almost done with this presentation. We have that butterfly shape that I had mentioned in a previous recording, and you can see it's very, very distinctive. The spinal cord has a central canal, a little bit of fluid in the middle, very tiny, an area of gray matter that is in, that canal is embedded in, and on either side of it, it has somewhat most anatomists described as a butterfly shape. Toward the back, it goes all the way, the gray matter, almost totally to the edge, where in the front, ventrally or anteriorly, it does not. That's how you know that this is posterior if you didn't have the bones around it, and this is anterior. And white matter is divided into what are called columns, and we actually can map them out. And in my advanced anatomy class, we do talk about how they're mapped out. And the gray are called horns, and they're also sensory inputs coming from the back, motor outputs go out in the front. And then these are the little bits of nerves that combine. When we talk about a nerve, it's not just one single strand, it's multiple strands that sort of fold into one. And here you can see the meningeal layers. And so, and in my advanced anatomy, we talk about the connectors, the various areas, we map it out. Same thing with the white matter. We can map this out as well. That's what this is about. In blue, these are sensory tracts. So most of your senses come up on the outside and a lot on the back. These are famously called the posterior columns. Most of the general information you get, I mean, if you close your eyes now, you know you're sitting. You know you're resting your face on your hand. You have your hands folded in front of you. Your head's pointing down without having to look, because all the information is coming from here. Pain, hot, dull, sharp, maybe you feel something uncomfortable on that wooden chair. That's called the lateral spinothalamics here. Big motor impulses are mostly here. The ones basically telling your muscles what to do. They're the corticospinal tracts, particularly the lateral ones. And then that's everything else about how we can predict what kind of damage someone would have if they've had a spinal cord injury or if it was cut, different diseases that affect it, etc. So that's why I have to share with you that. I'll take a couple of minute break so I can just check in, and then we'll set up for the dissection. So I'm going to stop this recording now. Guys, take a 5 or 10 minute break, and I'll set up for the dissection.